Okay, everyone, we're going to get started. My name is Greg Penny, and I'm the Director of Programs here at the Canadian Public Health Association. So we'll just do some housekeeping and a few items, and then we'll get into our presentation with Dr. Matthew Hill. So this webinar is being broadcast in listen mode only. There is the chat function that will be throughout, and we will also have a formal Q&A session after Dr. Hill's presentation. We are recording the webinar, and we will send the link to you and post it on our YouTube channel. So just for those that have not participated in these webinars so far, this is a webinar series as part of CPHA's project, A Public Health Approach to Cannabis and Other Substances, Prevention, Health Promotion, and Capacity Building. The purpose of our project is to engage a range of individuals and organizations from health, public health, social services, public safety, pretty much anybody working on cannabis, to enhance their knowledge of and capacity to address issues related to cannabis and other substance use. So this webinar is part of, as I said, our series that focuses on a range of issues related to cannabis consumption. I think most importantly, I'd like to draw your attention to the tagline at the bottom, which our focus is really on normalizing conversation, not consumption. And as this is a series, we do have another presentation coming up in, not April, sorry, wrong date, in May. My apologies. I realize after all this, I just noticed. So that will be taking place in May, and I will send that out properly. It's May 17th. I'll get to introducing Dr. Hill here, but I'd like in the meantime for you to actually focus on these polls that are here. So Dr. Matthew Hill is an associate professor in the Department of Cell Biology, Anatomy, and Psychiatry at the Hotchkiss Brain Institute at the University of Calgary. Dr. Hill is the former president of both the Canadian Consortium for the Investigation of Cannabinoids and the International Cannabinoids Research Society, and is the recently elected vice chair of the Gordon Research Conference on Cannabinoids in the CNS. A large portion of Dr. Hill's work has focused on the endocannabinoid system, the brain's natural version of cannabis, similar to how endorphins are the brain's natural version of morphine. The Hill Laboratory has found that endocannabinoid signaling in the brain largely acts to dampen the effects of stress. They have also demonstrated that stress hormones drive an increase in endocannabinoid activity in the brain, and that this increase in endocannabinoids is required to end the stress response. His research has demonstrated how chronic stress can compromise endocannabinoid function, identifying this system as target for both pathophysiology and treatment of stress-related mood and anxiety disorders. He has published over 100 papers in the field and has sat as guest editor on special issues of Neuroscience, Neuropsychopharmacology, and the International Review of Neurobiology, all of which focus on the biology of the endocannabinoid system. Dr. Hill holds a Tier 2 Canada Research Chair in Neurobiology of Stress, was the 2016 Canadian Association of Neuroscience Young Investigator Awardee, and was recently elected as the member of the Royal Society of Canada. He has worked with nonprofit, corporate, government, and academic entities to promote knowledge translation and communicate science to the public, and has been featured in venues such as CBC Radio, Scientific American, and the New York Times. And with that, I will turn the presentation over to Dr. Hill.
Thanks a lot. Okay, so just to give everyone a little bit of orientation, I think what I'm going to do today is just kind of review the basics of cannabinoids. I call this talk kind of Cannabinoids 101, as I just kind of orient everyone to the basics of the biology of the constituents from cannabis that can influence the endocannabinoid system, as well as give you a little bit of a background on the endocannabinoid system. Okay, so as is always the case, I think defining things at the outset is very helpful especially for a field like this where there's a lot of overlapping terms that can often be confusing. And I think a lot of this comes down to how we define what cannabinoids are. And so in the field, we always look at, there's two kinds of cannabinoids. There's exogenous cannabinoids, and that means these are molecules that are not produced within our body, but things in which people consume or take into our body. And there are the endogenous cannabinoids, which are the ones our body makes. I'll talk about the endocannabinoids in a second, but I'll start with exogenous. So from exogenous cannabinoids, these kind of come in two flavors. The first of which are phytocannabinoids, and what that means is these are actual cannabinoid molecules that are derived from the cannabis plant themselves. So the ones that are the most widely known certainly is THC, because that's the psychoactive constituent of cannabis. But in addition to THC, there's probably around 70 odd other cannabinoid-like molecules that are in cannabis. The second one, which has probably received the most amount of attention, is cannabidiol, or more widely known as CBD. So anything that comes or is derived from the cannabis plant is called a phytocannabinoid. The other type of exogenous cannabinoid that exists are synthetic cannabinoids. And again, this term probably requires a little bit more broadening. And I think one reason why I would even highlight this is because I just saw a recent debate on Twitter about this, because the term synthetic cannabinoid carries, I'd say, a lot of emotion with it to some degree. And a lot of that is because most people in the general population think when they hear synthetic cannabinoid, they think of things like K2 and spice, and a lot of these other ones that are hitting the streets right now that are being made in labs and are kind of these synthetic cannabinoid agonists. Now, there's clearly been demonstrated a fair amount of health risk associated with these. There have been documented mortalities from cardiovascular responses. So these are generally very adverse negative compounds. But the problem is there is also a medical therapeutic version of quote-unquote synthetic cannabinoids. And these are things like nabilone, dronabinol, marinol. These are all basically synthetic forms of THC. Now, the reason I highlight that it's important to define the differences between these is because I didn't realize the way that the public perception was around this until I was talking to someone about how they had issues with insomnia. They were asking about they didn't want to use marijuana. So I said, well, you should talk to your doctor about getting a prescription for nabilone then to try that. And they said, what's nabilone? And I said, it's a synthetic cannabinoid. And they kind of freaked out, saying, oh, but you know, I've read in the paper about synthetic cannabinoids resulting in death. And so I've started to realize that we really need to break these up into perhaps calling these pharmaceutical grade cannabinoids as opposed to synthetic, so that people understand the difference. But from a chemistry perspective, they're all synthetic. So it's important to understand that there's a difference between those. We also have our own kind of endogenous cannabinoid system, and this is the receptor system that THC and some of the other cannabinoids act on in the brain and body. But these receptors exist because we have our own endogenous molecules, or what we call endocannabinoids. And these are, I think the easiest parallel here is to liken these to endorphins. So the same way that our body makes endorphins, they act on mu-opioid receptors, which is the same target of morphine. Endorphins are endogenous morphine. That's where the word comes from. It's a conjugate of that. So endocannabinoids are very similar in the sense that they are molecules synthesized and released by cells in our body that act on the same receptor that THC does. Now, as we've learned certainly in recent time, that there is a very big difference between endocannabinoids and THC, the same way there's a very big difference between endorphins and morphine. They may act on the same receptor, but the way the body metabolizes these molecules is very different, and they have very different kind of lifespan of actions, let's say. Now, within the cannabinoid system, the receptor targets, and by receptors, these are the kind of protein complexes that cannabinoids like THC or endocannabinoids bind to elicit a cellular response. There are two receptor targets in the cannabinoid system we know of. There's the cannabinoid type 1, or CB1, and cannabinoid type 2 receptor. The cannabinoid type 1 receptor is really the main one that research is focused on, because within the brain, that's the primary receptor in the brain, and it's the one that we know mediates the psychoactive effects of THC. And we also know it has profound effects on neurotransmission within the brain as well. Okay, so targets of phytocannabinoids. So as I said, there's kind of two main cannabinoids that are being studied. There are two main cannabinoids in cannabis that are studied. There is THC and CBD. And you can see the structures here. Now, structurally, these molecules look somewhat similar to one another, but they have very, very different biological effects. Specifically, CBD does not act at cannabinoid receptors. I'll talk a little bit about the pharmacology of CBD as we proceed. But THC is the one that has received the most amount of research because it was identified as the psychoactive constituent of cannabis quite some time ago. Okay. So we'll focus a little bit on THC because this has been what has given us the most insight into the cannabinoid system. So ironically, the original publication of THC, which was discovered in the early 60s, was actually published on April 20th, 1964. There's a lot of, let's say, rumor and speculation that this publication date has in part spurred the pop culture referencing of 420. I do find that hard to believe, just because that would require a certain amount of knowledge about kind of old school publications. But it was discovered by Raphael Mechoulam. He was at the Hebrew University, and through a series of chemical isolations of different compounds within cannabis, he was able to isolate that THC was the psychoactive constituent of cannabis. So once this had been isolated, this kind of was a very major step forward in the field because it then allowed us to begin trying to understand how it was that THC and cannabis, let me see, I'm just trying to see, can everyone hear me? I'm seeing comments that there's some difficulty, so I'll turn my volume up. Can you tell me if that's better? Okay, I'm going to assume that that's better. Okay, so this kind of spurred the field forward that trying to understand the biological mechanism of how it was that THC was affecting the brain and producing physiological and psychological effects. So this led to the discovery of the endocannabinoid system. And essentially through a series of studies, mostly which were conducted in the 1980s and into the early 90s, it was discovered that THC exerted its effects through activation of an endogenous receptor complex system. So we referred to this as the endocannabinoid system. And so what we know is that there are two kinds of receptors that I mentioned earlier on that THC binds to exert its physiological effects. When we talk about the psychoactivity and the way that THC modulates the brain, this is almost entirely mediated through activation of CB1 receptors. CB2 is the second type of receptor I'm going to talk about briefly towards the end just because I think it's relevant for understanding this in the context of inflammatory disease, and these are found in immune cells. So CB1 receptors are found primarily in the brain. They are also found throughout the body, but this figure just kind of illustrates the differences in the distribution patterns. Most CB1 is in the brain. CB2 is mostly in immune cells, circulating white blood cells, spleen, tonsils, things like that. So when we talk about what THC does in the brain, what we're really looking at is how THC activates cannabinoid CB1 receptors. Now, obviously, there's going to be some variety in terms of the background of what people have for neuroscience here, but basically to explain this in a nutshell, brain cells, neurons in the brain, talk to one another through the release of chemicals. So one of them, which is we call the presynaptic neuron, this is the neuron that releases chemicals into the synapse, activates a postsynaptic neuron by those chemicals acting on receptors. Now, these presynaptic neurons can either release chemicals that are excitatory, so they activate postsynaptic neurons, or inhibitory, so they dampen postsynaptic neuron activity. Now, cannabinoids, and where the cannabinoid receptor is kind of different than the way that most neurotransmitter systems work because the cannabinoid CB1 receptor actually sits on the presynaptic side as opposed to the post. And what that means is when that receptor becomes activated by THC, it doesn't directly influence neural activity in those postsynaptic neurons, but what it does is it modulates the amount of transmitter that gets released into the synapse. Now, these receptor systems are kind of distributed widely throughout the brain, and they're found in a lot of different centers of the brain that regulate different physiological processes. This does give us a fair amount of insight looking at what the kind of physiological and prototypical effects of cannabis consumption are as to what that means the endocannabinoid system itself probably regulates. So one of the more prototypical effects of cannabis is this shift in feeding behavior. So people consume cannabis and they want to eat sweet, palatable, tasty foods, or what people kind of colloquially refer to as the munchies. This was one of the first things that the field kind of jumped on once the endocannabinoid system itself was discovered. And what was found was that similar to the way that THC causes increases in feeding, there are actually cannabinoid CB1 receptors distributed widely throughout feeding centers in the brain. And when that receptor becomes activated, it favors the engagement of feeding behavior. And so what the field learned was actually endocannabinoids themselves get released during various states, such as fasting or a period of food deprivation. Endocannabinoids are released into these feeding circuits in the brain, and when they activate CB1 receptors, they try and promote feeding behavior to replenish caloric content and eat sweet palatable food that's high in calories. So essentially what you can look at this as is, if you think of a natural physiological process like feeding that endocannabinoids might regulate, what happens when someone consumes cannabis and THC begins to indiscriminately activate CB1 receptors in these feeding circuits, in some ways it's tricking the brain into thinking that the individual has undergone a period of fasting and therefore needs to eat again, and that's why it triggers that kind of munchie-like behavior where people want to eat high-calorie foods. Now, using that logic, we can look across a whole slew of different physiological processes, and most of them seem to have the same effect. And another one being stress and anxiety, which is an area that I work on. And again, this is something we've learned over the last 15, 20 years, that endocannabinoids are released in emotion circuits throughout the brain, usually in response to stress, in an attempt to help calm our brain back down and return to normative function. And this is likely one of the reasons why the majority of individuals who recreationally use cannabis on a regular or even daily basis, when you look at the self-report claims, the main reason why they do so is because it reduces sensations of stress and anxiety. And again, this is likely because of the distribution of these cannabinoid receptors throughout emotion circuits in the brain, and when they get activated by THC, it kind of triggers this relaxation response because it's part of a normative stress recovery process. And lastly, the other one that has certainly a lot of attention in the medical community is the relationship between cannabis and pain, because the number one reason why medical populations use cannabis is to manage chronic pain syndromes, especially neuropathic pain. And so similar to how the endorphins, when they were discovered, it was found that they are released in response to painful, noxious stimuli and act to almost an endogenous analgesic. It looks like endocannabinoids actually do a very similar process. And what I mean by that is, in response to a painful stimulus, it does appear that endocannabinoids are released, and that mediates the sensation of analgesia. Other things such as exercise and heavy levels of physical activity, they also seem sufficient to mobilize levels of endocannabinoids, and that release of endocannabinoids in response to exercise does seem to mediate some of the kind of analgesic and rewarding effects of running. So it's important to that helps put things in context in terms of understanding the endocannabinoid system and how cannabis influences it. Okay, I'll just do a little bit of a sidebar on anxiety, because this is an area that I know very well, and it can kind of be used as a case to get an understanding of how cannabis interacts with the endocannabinoid system to influence a process that could possibly involve a pathology. So as I mentioned, endocannabinoids are naturally released in the brain to promote recovery from stress. They reduce anxiety, and they also help to dampen rumination, or when people perseverate and go over and over again emotionally aversive or negative memories. So the endocannabinoid system acts to counter a lot of these processes and favor stress recovery, reduce anxiety, and kind of clear negative memories out of the head, and almost a forgetting process. Now, not surprisingly, it's quite interesting that there's a high degree of cannabis use in PTSD. Now, there haven't really been really well-controlled clinical trials in this domain. There's been a few that have come from the Canadian military that have looked at nabalone in isolation, so just THC administration in the context of PTSD, and have seen some benefit, particularly with respect to sleep and nightmares in PTSD. But on some of the clinical work that we have done in collaborating with researchers who study PTSD, we've looked at endocannabinoid levels to see if there's an alteration in the system in a disease state like PTSD that is accompanied by high anxiety and inability to recover from stress and have these kind of persistent negatively emotionally aversive memories. And so through a series of both kind of clinical and animal studies, the hypothesis that we've come up with for the most part is that some disease states, for example, PTSD, may be associated with impaired or deficient endocannabinoid activity. And in these situations, the individuals who are self-medicating may be using THC or cannabis in an attempt to compensate for a deficit in the normal functioning of the system, and thus bringing it kind of back up to a normative level and helping to reduce a lot of the symptoms associated with the disease. Researchers who work in the pain field have come up with a similar model for chronic pain syndromes, where they found that individuals who either have chronic inflammation or chronic pain may indeed have some compromised endocannabinoid functioning, and that may be one of the reasons why they're more vulnerable to develop certain disease states. And a subset of these patients, when they try using cannabis, may actually find benefit from it because it could be restoring normative function. And so this area requires a fair amount more work to really kind of fully flesh out and understand what's going on here, but this is the model that we've used to try and understand why very specific disease states, such as things like pain and PTSD and anxiety, which we know are processes that are normally regulated by endocannabinoid function, may have a high degree of self-medication in individuals who are using cannabis. So this is just kind of a snapshot based on a large series of studies that have tried to understand where cannabinoids are acting in the brain to modulate a whole host of different physiological and neurological processes. So one of the things that's very interesting about the cannabinoid CB1 receptor is it really is found almost everywhere in the entire brain. The one area it is notably absent, and this is very important and interesting, is in the respiratory and cardiovascular centers of the brain. And the reason I point that out as being extremely relevant is that is where there is an abundance of opiate receptors, and it is activation of opiate receptors in that part of the brain that regulates heart rate, cardiovascular function, and breathing that causes fatalities through overdose, because when opiates activate their receptors in that part of the brain, individuals tend to lose consciousness, but in the process their heart rate slows down and their breathing rate slows down to a point where they end up dying. And so the fact that there are no cannabinoid receptors in that region of the brain explains why there's never been a reported fatality associated with cannabis use in human history. And it's interesting to note that the fatalities that have recently been associated with synthetic cannabinoid use have almost all to do with the heart and are usually related to cardiac arrest. And that's because there are cannabinoid receptors in that area, but for some reason, because of the biology of how THC acts, it doesn't seem to promote these same effects. But synthetic cannabinoids, like K2 and spice and some of these other ones, have very different pharmacological properties that actually make them far more dangerous because of the way they activate the receptor. They can trigger these cardiovascular responses. But as you can see, just from surveying this, you see there are receptors in the cerebral cortex where it's going to affect processes like a decision-making and cognition. There are areas like the caudate nucleus and the putamen where it's going to influence various forms of learning. The ability of cannabinoids to kind of impair short-term memory. So a lot of individuals who are under the influence of cannabis, it becomes obvious that they have some deficits in short-term memory function. That appears to be more mediated by an area referred to as the hippocampus. For the stuff that I study, anxiety, stress, emotion, fear, the amygdala is a very important part of the brain for this, and it's also, interestingly, a part of the brain that is hyperactive in individuals that have PTSD. And there's been some clinical work in humans that show administration of THC can actually blunt neural activity in this brain region. So this is just kind of, as I said, a snapshot of seeing how the diversity of both regions and actions that cannabinoids can influence on brain function. Okay, but as we know, as I alluded to at least, CB1 receptors are also found outside the brain, and they're found in almost all tissue systems in the body. And this can actually have a lot of functions that are important both from a potential therapeutic benefit, but also from a potential damage perspective. So areas such as the pancreas, which are involved in insulin release and therefore glucose regulation, there is kind of mixed reports on how cannabis or cannabinoids can influence blood glucose levels that could either be potentially beneficial or damaging depending on the type of diabetes an individual would have. So that's something that is getting a lot more research now. We know that cannabinoids can act in the liver to modulate a lot of processes like lipid storage. There is some accounts of individuals who have liver diseases where cannabis probably should not be used because of potential interactions on the liver, such as hepatitis. There is definitely effects in adipose fat tissue where endocannabinoids may influence the development of fat tissue to some degree. Same with muscle, it can influence glucose uptake in muscle. And I think importantly, the gastrointestinal system is one we've really started to learn has a lot of cannabinoid receptors that could be heavily influenced by cannabinoid actions. And this is really important for things like colitis and inflammatory bowel disease, where there's a very high degree of cannabis self-medication. Okay, so the other kind of receptor I mentioned before is the CB2 receptor. So the CB2 receptor is the other cannabinoid receptor. It's the one that has a lot less attention and a lot less research focused on it because it's not really in the brain and it doesn't really influence neural processes the same way. It really is primarily found on immune cells. And by what I mean by immune cells is, as I mentioned, kind of circulating white blood cells or macrophages, but also things like the spleen and the tonsils. And what's interesting about the CB2 receptor is when it is activated by THC or by endocannabinoids, what it acts to do is actually suppress the release of inflammatory molecules. Now, what's really interesting about the way that the system works is it doesn't seem to do much to a normal immune system. Now, the reason this is important is because in the early days when they discovered that cannabinoids could influence immune processes, the original thought was this actually cannabinoids might induce immunosuppression because they'd be reducing the release of inflammatory molecules from blood cells. Now, this was of particular relevance because some of the very early studies that were looking at medical uses of cannabis were looking at it in the context of AIDS, where an individual is already immunocompromised. And so in that situation, they wanted to make sure that they weren't actually making anything worse, and they weren't making an individual more vulnerable to the immunosuppressive effects that were associated with HIV and AIDS. And the reason for this was because cannabis was being used to try and counter the wasting syndrome or cachexia that's associated with HIV. So the original studies looked at this, and quite to their surprise, they found that cannabis had no influence on immune function in someone who was already immunocompromised. Similarly, there seemed to be almost no effect of cannabis on immune function in someone who was relatively healthy. But what was interesting was someone who seemed to be suffering from an inflammatory disease where the system was hyperactive. Then you saw this effect of cannabinoids to be able to reduce the release of inflammatory molecules. We now know that this is likely due to the fact that at rest, these immune cells don't really express many CB2 receptors. And what happens is when an inflammatory event happens and these immune cells become, let's say, angry and start dumping out mass amounts of inflammatory molecules, they also start expressing cannabinoid CB2 receptors. And that allows them to have a mechanism to turn themselves back off. And so in many of these chronic inflammatory conditions, we think the reason why cannabinoids may have some therapeutic benefit is because they would have the ability to activate CB2 and curtail the release of inflammatory molecules to kind of prevent the propagation of these disease states. But again, it's important to know this really only seems to be true under conditions where the immune system is hyperactive, not when it's at rest or when it's compromised. Okay, I'll quickly mention cannabidiol or CBD just because this is something that everyone seems to be talking about nowadays. This is a mystery drug. We don't really know what this drug does in terms of its mechanism of action. And it's been an area that's getting a lot more research now because of the fact that there's so much kind of widespread use of CBD. What we do know is that it does not directly bind to CB1 or CB2 receptors, so it's not a true cannabinoid in that sense. What it does seem to do is it has an influence on a whole host of other neurochemical systems. So one thing it seems to do is inhibit adenosine uptake. And so adenosine is a molecule in our brain that acts to kind of quiet down a lot of processes in the brain, and it helps to promote things like sleep. So caffeine, for example, has the opposite effect of CBD. Caffeine impairs the actions of adenosine, and that's one of the reasons why caffeine makes us so alert, is because it compromises adenosine signaling in the brain. CBD, on the other hand, promotes the actions of adenosine signaling in the brain. So in some ways, you can almost think of this mechanism as being kind of an anti-caffeine. And so that's one of the reasons why a lot of people seem to report an improved ability to sleep or relax with CBD, even though it doesn't seem to have frank psychoactivity because it doesn't seem to be activating CB1 receptors. We also know that CBD can interact with serotonin receptors. This is similar to a lot of drugs that are used to manage symptoms of depression and anxiety interact with the serotonin system. And so, again, there has been some reports, although they haven't been entirely consistent, that CBD has some anxiety effects. And the thought is that this is probably the system. In terms of the effects of CBD, the most reliable effect that seems to be documented today, certainly in a medical context, is the anti-epileptic effects. And this isn't broad across all forms of seizure disorders or epilepsy, but it seems to be very specific to these types of childhood epilepsy, such as Dravet syndrome. And in these diseases, there's now been one published and I think 2 to 3 trials that have been completed and shown positive effects, but not published yet. But they will be out soon. all demonstrating that CBD seems to be able to reduce seizures in a lot of these kind of rare forms of pediatric epilepsy. And that's very interesting, and I think there's going to be a lot of research pushing that direction. The anti-inflammatory actions of CBD have also been documented, as well as possibly some anti-anxiety, but most of these haven't been done in human populations and do require a bit more work. Okay, so what do we know about how cannabis affects the brain? This is kind of coming towards the end of the talk now. I'm just going to briefly explain where a lot of the discussion points that have made it into the media and onto the political arena about the effects of cannabis on the brain have come from, and what we understand about them and how we need to improve them moving forward to understand things better. So the first one is human epidemiological studies. And so in these studies, when they've tried to look at the effects of cannabis on specific outcomes, they look usually in a retrospective manner. They look at comparing populations that either smoked cannabis or didn't. Now, obviously for many things, this kind of approach is a good way to get an idea if there's a relationship between two variables. The one problem that's become very apparent in the realm of cannabis, however, is that for one, the groups are often poorly controlled in terms of their relationship to one another. And so, especially in the United States, a lot of the groups that are studied who use cannabis are almost entirely marginalized parts of the population. They usually live in very low SES. Individuals who started using cannabis at a very young age, for example, tend to have a lot of other issues in their home life or their personal life that could influence a lot of the outcomes that are being measured that therefore may not be directly attributable to cannabis. This is important because as studies have gone back to try and replicate some of these kind of media splashing headline studies, for example, the IQ one that tried saying that teenagers who consumed cannabis had this persistent decline in IQ throughout their life, it has subsequently been found that that actually was not related to cannabis, it seems. It's related to other variables that track with cannabis use. For example, that study, groups have gone back and done twin studies looking at identical twins where one smokes cannabis and one doesn't, and find similar declines in IQ, suggesting that there's either some pre-existing differences or some other functional variable that mediates the relationship between these two. So this has made it very difficult now for us as a field to look back and make firm claims about relationships between cannabis use and specific outcomes. The other way that we've learned things about how cannabis or cannabinoids influence the brain is through animal studies. Now, this is much more in my wheelhouse and stuff that I'm familiar with, and some of the problems that are very apparent in this field now are the fact that the dosing of the drugs that are being used and the route of administration is not reflective of reality. Most animal studies use injection-based approaches of drugs. This is usually to try and control exact dosing levels, and for many things this makes sense, but for cannabis this really doesn't make sense. And we know this now because there's been a lot of research on the pharmacokinetics of cannabis, and we understand there's a very big difference of the biology of this, of how this plant and its molecules interact with the body if it's smoked versus if it's eaten. And injection is probably going to look a lot more like oral consumption, like eating it. And that's just because of the fact that it goes through the liver and it's metabolized and it produces different molecules than what happens if someone smokes or vaporizes it through a pulmonary route and it goes in through the lungs and gets direct access to the brain. And the reason this is important is because the majority of adverse outcomes that have been associated with cannabis in terms of especially the subjective effects are far more common in individuals who've consumed cannabis through an oral route as opposed through a pulmonary route. And a lot of this has to do with the way that the body handles THC and metabolizes it. And so these animal studies are using very high doses of THC quite often. They're injecting it, so it's having very different effects than what we're seeing through what would occur through kind of regular patterns of smoking or vaporizing. The other main problem we've seen in the animal studies is that they tend to only study THC or even synthetic cannabinoid agonists as opposed to cannabis as a whole plant. And as we've started to study the other constituents of cannabis in the last few years, such as CBD, we've really started to recognize that there's an interaction between these systems. So there's a high level of CBD. It seems to be able to counteract some physiological effects of THC. And that's the only one we've really studied in an interactive manner. And so now we're starting to realize that as much as scientists, we love to do reductionist approaches and study molecules in isolation. For something like cannabis, if we really want to understand what it's going to be doing to the brain, we need to start studying the impact of whole cannabis. And so rodent models studying this are going to have to move into this direction. And there are several labs that are starting to do this, and I think the data that will come from that will be far more informative. And so what we need to do moving forward to actually be able to come up with more definitive answers about what we understand about how cannabis is going to influence the brain is restructure our approaches for both animal and human studies. And this is being done now, as I alluded to. And the first one is the way that they're doing these longitudinal human studies. So in the United States, the NIH, primarily the Institutes of Mental Health and Drug Abuse, have collaborated on this very expensive, large-scale 10-year study called the ABCD, or what is an acronym for Adolescent Brain Cognitive Development Study. And what they're doing is they're tracking about 10,000 kids from the age of 10 to the age of 20. And they're going to be doing baseline neuropsych, baseline neuroimaging, baseline IQ, figure out as much as they can at the beginning, and then track these kids throughout adolescence. Look at the ones that begin using drugs, explicitly the ones that begin using cannabis, and track them and try and understand if any effects that have been seen with cannabis through these kind of retrospective studies are indeed noticeable in real time. Can you see trajectory differences? If someone who is on one path begins using cannabis, does that alter their trajectory? And if so, is this true only if cannabis is used in combination with other drugs? Is it true if the individual has a certain history of life stress or low SES? Is this true if it interacts with other biological variables such as genetics? And all this data is being collected, and the sample size will be large enough that the hope and the goal is, from doing longitudinal studies like this, we really will have far more detailed information about the relationships between cannabis use and specific outcomes. From the animal study perspective, we really need to start changing the route of administration. We need to start modeling inhalation as the main approach, since this is how about 90% of individuals who use cannabis recreationally and to some degree medically consume it is through either vaporization or smoking. Now, vapor chambers have been developed for animal research for some time, because that's how they've been studying cigarette smoking and nicotine for years now, and so a lot of these are starting to be co-opted. And the important thing about using a model like this is that you can begin to look at differences between things like high THC, high CBD. You can begin to titrate blood levels of animals so that the amount of THC in their circulation matches what a human who consumes cannabis through smoking a joint would. And so therefore we have a lot more valid translational approaches. And the other approach by using these vapor chambers is we can start using either the whole cannabis plant or whole cannabis extracts and oils. And that way we're not just looking at the effects of THC in isolation or CBD in isolation. Doing those studies is obviously important to understand how THC in and of itself can influence the brain, but understanding it in the context of how the whole plant influences the brain, especially in the context of the two main questions right now being prenatal development. If moms are exposed to cannabis smoke or cannabis vapor while they're pregnant, what does that do to the developing brain of the fetus? But the other question is, what does that do to the developing adolescent brain? Because there's been a lot of speculation about whether or not cannabis use during adolescence has these profound effects on brain development. Now, there is some data to suggest it might if it's excessively high use of high THC cannabis, perhaps, but there are also some studies that have used very, very well-controlled groups that have even looked at teenagers that smoke cannabis on a daily basis and not found any effects on the brain structure in individuals who smoke cannabis daily. And even that approach still works if you're trying to find a signal with alcohol. So it's not as if being very careful with controlling your variables would remove the ability to detect a signal. It's just, it has brought into question whether or not it's cannabis in and of itself that is impacting the development of the teen brain versus cannabis coupled to heavy alcohol use, heavy cigarette use, heavy life stress, some underlying genetics or predisposition to certain mental illnesses. Okay, so I guess the main question at the end is really, should we be worried? And you know, there seems to be a lot of varying opinions on this, and so I'm just going to point out two things that I think are relevant. Now, the first one is this kind of allusion to the Reefer Madness movie from the 20s or 30s when this came out. And I say this because, as someone who works in kind of a field of biological psychiatry, the relationship between cannabis and schizophrenia and psychosis has become a very, very hot topic. This has trickled into Parliament Hill, and has obviously made its way into the discussions of the senators there as they kind of go back and forth about what people expect to see in terms of negative effects from cannabis legalization. So there is clearly a epidemiological relationship that exists between cannabis use and schizophrenia. Schizophrenics do seem to have much higher rates of cannabis use than the general population. That's not entirely surprising since schizophrenics do also have substance use and abuse rates much higher than the general population, but there is a very interesting relationship that exists there with cannabis. There is evidence that individuals who start smoking cannabis at a very young age likely have an earlier onset of the disease, and if they continue smoking cannabis regularly through the disease, their long-term prognosis may be worse. But there is a very emotional debate, let's say, that goes on in the field about whether cannabis itself causes schizophrenia or psychosis. Now, we do know that under acute conditions, if someone consumes a high amount of cannabis or a high amount of THC, this often happens through people using edibles where they aren't able to titrate what they're consuming appropriately. They may indeed have a transient psychotic reaction, which usually ends them up in the ER. This is usually characterized by a profound panic attack. They feel like they're detaching from their body. They don't know what's going on, and they often just need to be sedated for the night, and in the ER in the next day or so, they tend to normalize back down to baseline function. There are a subset of people, however, that seem to have a psychotic episode triggered by using cannabis that then turns into a long-standing form of schizophrenia. Now, the debate has been whether this is something that could just happen from any old individual smoking cannabis developing schizophrenia, or if this is more so that cannabis can act as a trigger to bring out schizophrenia in individuals who are vulnerable and would likely already develop the disease. Now, I'm of the perspective that correlation does not equal causation, and when we look at large-scale data about schizophrenia rates across time, which granted there are some issues associated with data collection, despite the fact that we see very big differences in cannabis use rates in teenagers across the world. For example, Canada has essentially the highest rates of cannabis use in teenagers, with some studies suggesting by the end of high school, 1 in 2 teenagers have smoked pot. Yet we compare our schizophrenia rates with other countries such as the Scandinavian countries like Norway, where cannabis use is very, very low and almost non-existent, where somewhere between only 2 to 5% of teenagers smoke pot. And our schizophrenia rates aren't really that much different. Similarly, we look historically kind of pre-1960s to post-1960s when cannabis became introduced into Western culture. We really have not seen very much shift in the prevalence or diagnostics of schizophrenia, which to me argues against there being a causal association, because it's difficult to reconcile the fact that cannabis use has gone up dramatically, especially in countries like Canada, yet our schizophrenia rates have remained relatively stable. So I think realistically what we're looking at here is that cannabis use for individuals who are biologically predisposed. Now, these people often won't know that they are because there's no test. We have no ability to know who's going to develop schizophrenia and who won't. May be very sensitive to the adverse effects of cannabis, and it may help bring out the disease. But that is a fundamentally different interpretation than there being shifts in actual prevalence rates of schizophrenia. So when we talk about this in the context of legalization, I think the first point to mention is very important is we already have very high rates of use of cannabis use in adolescents. And the other point is we have absolutely no reason to assume that that's going to change post-legalization, because despite the fact that with older age groups we do see some small increases in cannabis use following legalization, for teenagers, all the data that has come out so far from Colorado and Washington has not demonstrated any use changes in teenage population groups following legalization. And Deborah Hassan, who's an epidemiologist at Columbia, has done very careful analyses across this from states that have no legalization versus medical access versus recreational legalization. She has not been able to detect any signal in high school age children and teenagers that they have any changes in use patterns following legalization. So that's an important thing to recognize, because it does suggest that we probably aren't going to see a shift in that population. And if that population is who we're highlighting as one of the potentially vulnerable populations, it's important to establish that there's really no reason to expect a change in use in that group. So my take-home message overall from this would be, I don't think that we should, for these reasons at least, I don't think we have any reason to necessarily be concerned, because I don't think that we're going to start seeing dramatic changes in prevalence rates of mental illness. I think that that's a misinterpretation, I would say, of epidemiological data showing correlations between cannabis use and mental illnesses. And also, I just don't think that there's any evidence to support the fact that teenagers are going to start using more than they already do. I think there are certain things that we do have to be aware of moving forward with legalization that will be kind of concerns in the public health domain, and this could have to do with the fact that even if a small proportion of people do start using cannabis more, we're really going to need to track rates of how much cannabis an individual uses, because we may see that more individuals shift into problematic use patterns, even if the gross prevalence of cannabis use itself doesn't change. And so we will need to have some very good baseline data. There has been some collected in the country. Hopefully this will be aggregated into a larger data set for us to be able to collect and track these variables over time. And that's the end of my presentation, so I will be happy to field questions for the next 15 minutes.
Great. Thank you very much, Dr. Hill. And so as he mentioned, we have about 10, 12 minutes for questions. So what we'll do is there's two areas to put it, both in the chat and in the Q&A, and we will sort of get to as many as we can. So Dr. Hill, actually two just points of clarification. The last point you're making around some of the data and youth, is that data from the US or Canada? And the second point is further back. Yeah, go ahead.
Okay, sorry. I was just going to say, yeah, I'm just answering the Q&A right now. For the figure that I have on the screen right now, that data is from Colorado. So that is, and the inflection point there is that the end of 2013, beginning 2014 was when legalization occurred. So for the 2 years following legalization, when they tracked these numbers, there was no change in Colorado.
Okay, perfect. So there was an earlier question also about how CBD interacts with serotonin, simply because there was a drop-off. Can you just sort of quickly touch on that again?
So the CBD seems to be able to interact with certain serotonin receptors. It doesn't bind to the main binding site of the receptor. It binds to another portion of the receptor protein itself, and it can enhance the ability of serotonin to bind to those receptors. So because of the specific serotonin receptors that it's able to do this with are related to several anti-anxiety and some antidepressant medications, and because the animal work that's been done looking at anxiety-like behavior has suggested that the actions of CBD involve the serotonin mechanism, it's thought that that's probably how it's mediating those effects. Okay, great.
So this is sort of a maybe not something that is being asked right from my part. So is the skin a CBD receptor? And because obviously there's a lot of products that are topicals and balms, and so how does that actually affect the system, and how is it absorbed?
There are CB1 receptors in the skin. They certainly wouldn't relate to any kind of psychoactivity or anything. There's some suggestion that, I mean, it may influence things like oil secretion from sebaceous glands. There is some suggestion it could influence keratinocytes and influence wound healing, but no one's really looked at that properly. But when you talk about absorption, I think the idea of using these kind of topical cannabinoid-based products, that's going to be entirely dependent on the medium that they're in. I mean, the skin is a barrier, so it acts to keep as much as possible out. But a lot of these topical solutions come with a penetrant that's going to help pull the molecules through the skin. So the ability of those compounds to work, those topicals to work, is going to be entirely dependent not on the cannabinoid, but on the basis of what's in them. Because if they absorb well and if they do pull the cannabinoid with them, then they will get it into the system to some degree. I wouldn't imagine you'd be able to, at least unless it was extremely high concentration THC, if that would be able to influence psychoactivity, because I think the absorption rates are pretty low from anything I've seen.
Great. So you'd mentioned early on the endocannabinoid system and the receptors. Does cannabis have a cumulative effect? Is it held? So when people consume, is it held in the system, and is there a cumulative sort of buildup of cannabinoids in the endocannabinoid system?
It will store in the fat. So because THC and most cannabinoids are lipid molecules, they like fatty environments. And so what happens is once they get into the blood, they usually are bound to a protein that carries them around in the blood, because blood is like an aqueous solution, so lipids won't stay in it very easily. So what happens is once it gets into the blood, the main area it goes is not through metabolism. It's not like alcohol where you metabolize it and kind of excrete it back out. That does happen with THC and other cannabinoids, but it's very slow, because what happens first is they shift into the fat cells in your body. And so individuals who smoke cannabis on a regular basis, if they have a reasonable proportion of body fat, their body fat will store a fair amount of THC. And over time, as blood levels of THC start to decline, the fat will slowly release this THC back into the blood. This isn't in a manner that's going to have psychoactivity, but it is going to be in a manner that will influence drug testing, for example. And that's why individuals can fail drug tests for THC up to 30 days out of consuming it, because if it's stored in their fat and slowly released, they're still going to have a signal in their body. In terms of I wouldn't say there's any data to suggest that this kind of cumulative effect in the fat would have any influence on the brain.
Okay, great. Are you aware of any evidence around the effects of cannabis on persons with intellectual disabilities or other issues, or maybe ADHD or other sort of research in that area?
I've never really seen anything that's been done on populations of individuals with intellectual disorders. There's a lot of anecdotal stuff with ADHD. There hasn't been very solid clinical trials. There was one trial, I think, that was done in Italy last year that used a cannabis extract product called Sativex that's made by a company called GW. And it's kind of an oral mucosal spray where you spray it under the tongue and it absorbs into the body. And it's usually a mixture of THC and CBD 1-to-1, as well as a lot lower levels of other cannabis constituents. But in the study that they did in ADHD, they did find, it was very minor, but they did find a significant improvement in a few domains, such as, I believe, impulsivity and attentional capacity. And that was kind of it's not a sledgehammer by any means, but it's certainly the only clinical evidence that I'm really aware of outside of anecdotal reports to suggest that there could be an improving effect for cannabinoids in ADHD, but that's as much as I'm aware of.
Okay. So what is the psychobiology of THC-induced euphoria or the high?
I probably would separate the two of those just because certainly the effects of THC I would not uniformly call euphoric. For example, if we try giving them to animals, they actually find it aversive. They don't find it euphoric at all. Often animals will self-administer to not get THC because they tend to not enjoy it unless they get very small amounts through vapor administration. But it is clearly for a lot of people who consume it, it's rewarding and it's reinforcing, that's for sure. And that is just due to the fact that it engages the dopamine system, which is very important for signaling stimuli that have rewarding responses or could be viewed as pleasurable or positive. So there is some effect there that it interacts with the dopamine system, and that is likely what mediates the kind of rewarding aspects of cannabis. The high, I would say that, no one really truly understands this, but it's probably to do with cortical cannabinoid receptors and just the ability of how activation of cannabinoid receptors can modulate cortical function. So you get a little bit of a depersonalization effect, or you might have altered thought patterns, and that seems to be more what I would call the high, and that probably has more to do with how it alters cortical information processing.
So the next question is there any evidence about prenatal cannabis use and fetus brain development and any specific prevention messages to use?
There's an area that actually doesn't have a huge amount of work done in it. There's been 3 large-scale cohort studies that have been done over the years. None of them have really found anything overwhelming. Now, the one reliable thing that you see across all the studies, and this has been replicated in the animal studies, is low birth weight. So that does seem to happen reliably from pregnant moms consuming THC or cannabis. At a meeting I was just at, the chief scientific officer of the College of Obstetrics and Gynecology was there, and she basically said, we get signals here and there that there are effects, such as, there might be some alterations in executive function or attentional control in individuals who are exposed to cannabis in utero. But she called it, if there are stuff, I think the term she used was more silent or invisible because it wasn't presenting the way, say, fetal alcohol would, where you get craniofacial shift and you get frank alterations in intellectual functioning at a very young age often. So there hasn't been documented reports of that, but I think the messaging that anyone would use would just be, obviously, if someone's pregnant, there should be an avoidance of use as much as possible. What was interesting from what the the CSO for Obstetrics and Gynecology College had said was when they asked women, a lot of women reported using cannabis, because this is what I was curious and surprised at, was what the motivation was. It was almost all to regulate morning sickness. And the perspective a lot of women had was they were concerned about effects of the drugs that were being used as anti-nauseants, and they felt more comfortable using cannabis. I'm not really sure why. That probably relates to some degree of the normalization of cannabis in the population, but that seemed to be the main motivating factor for use.
Okay. So there is one question here. So if there are potential therapeutic THC effects on anxieties, why do we hear so much about why anxiety gets worse over time? Is it that self-medication persons are using higher THC doses through non-medicinal sources for their cannabis?
Yeah, if an individual is using cannabis excessively, especially high THC cannabis excessively, over time that's going to compromise their own endogenous functioning. So the endocannabinoid system itself may become compromised. That doesn't seem to occur with low levels of use, and that's probably why it depends on the pattern of use that some people have. So there is clear epidemiological evidence that individuals that use a fair amount of cannabis in an attempt to regulate their anxiety symptoms, over time that may become worse. Now that is definitely a reasonable concern and something that needs to be more built into education.
Okay. So I'm just going to ask one more question. I know people are...
Military who've used cannabis to try and moderate their symptoms of PTSD, or even looking at the clinical data, the main effects that people see is if dosing happens right before bedtime, and usually it's very low levels of dosing. And I think if people are attempting to use cannabis and it means to regulate anxiety or symptoms of PTSD, really the approach should be very low levels of dosing at nighttime, almost as like a sleep agent to help reduce nightmares. For PTSD especially, it seems to be important. I think for anxiety, things need to be worked out a little bit more. But because of the long half-life of THC, because it stays in the body for so long, there really shouldn't be a need to dose as regularly as some individuals do, and that probably relates to problematic use. But the interesting thing is the biggest predictor from what I've seen in the literature of whether someone will develop a dependency on cannabis is if they're using it to try and manage social anxiety. And that probably generalizes to other anxiety conditions, but just social anxiety is very cleanly definable. And in that population, people who use a lot of cannabis to manage their symptoms often develop problematic use patterns.
If you allow me one last question. So obviously one of the hot topics is impairment testing and cannabis. Any last thoughts?
Yeah, I mean, there are, so when they've done kind of like simulated driving, I'm assuming this is in the context of impaired driving. So when they've done simulated driving tests, they do see that individuals have reduced reaction time and some increase in lane weaving and some attentional issues, especially as the task demand increases. But what separates cannabis from alcohol, and there's a big study that was just done at the Center of Addiction and Mental Health, which it should be getting published soon from what I've heard, but they did a very large-scale study of this. And what they found was that, and this is consistent with everything I've read in the literature, is that there's compensation behaviors that individuals use with cannabis that they don't seem to engage in with alcohol. What I mean by that is there's almost a recognition that they're impaired, and so they start slowing down. They start engaging in a lot less risk-taking behaviors while driving. And so the net effect of cannabis impairment on accidents was very, very small in most studies that have been done. The estimated risk is it's about a 2 to 6-fold increase if someone is driving while intoxicated with cannabis. That risk is going to vary depending on how intoxicated they are and what their history of cannabis use is. But as a frame of reference, the legal level of alcohol increases the risk of an accident from 3 to 18%, or 3 to 18-fold, and texting increases it in someone who's sober by about 6-fold. So there is a risk that there is an increase in impairment, and there's a chance that they will get in an increased risk that they will get in a motor vehicle accident. But that risk in the larger perspective is less so than what's associated with even legal limits of alcohol or texting while driving. Now, this is important because Colorado and Washington just released their data in the last few weeks over accident fatalities over the last 3 years since legalization has occurred, and they found absolutely no change in traffic-related fatalities pre- and post-legalization, as well as when they compared to other states surrounding who did not legalize. And so there doesn't actually appear to be a signal there showing that cannabis legalization will result in an increase in motor vehicle fatalities. Now, there may be a small increase in non-fatal minor car accidents, that data is a lot harder to capture, and so no one's really reported anything on that. But based on the traffic simulator studies, one may predict that, but I don't think that this is a very massive concern we have to worry about following legalization based on everything we've seen.
Perfect. So I know we've gone just slightly over time. I want to thank Dr. Matthew Hill for presenting today and for all of you submitting your questions. We have recorded this webinar. We will be circulating a link to that to you, and this has some slides. And if Dr. Hill's okay, we will circulate his presentation. Thank you, Dr. Hill, very much. It's very informative. I think this probably could have gone quite a bit longer based on just the questions, and we really appreciate your time.
Okay, thanks a lot.
Thanks, everyone, and enjoy the rest of your day. And we will send you a link to the next webinar coming up in May. Thank you very much.